All right, kiddos, how are you? Uh, we're back. We're back from spring break, and it actually was kind of spring-like, which is unusual for our spring break since it's so early, and it's not spring. But anyway, so I'm going to pick up where I left off, and I am on slide 14. And on slide 14, in the previous discussions, we've talked about what happens with our heart up here. And we had said that we have the two atria are separated from the two ventricles by a fibrous skeleton right there. And so we had said that we have groups of cardiac muscles, and as we call each of these groups a syncytium. And so we have the atrial syncytium and we have the ventricular syncytium. And we had said, how does the heart pump? It goes lub-dub, right? And other things we had said was that the cardiocytes, the cardiac muscle cells, we said they communicate. They talk. Yeah. And when we talk about communication between cardiac muscle cells, what we're really meaning is that they share their chemistry, right? So they share their chemistry, and that means when one starts to contract, it spreads to the neighbor, and then it causes him or her or it to start contracting, and it spreads to their neighbors, and so on and so on. It happens very quickly, though, but it spreads throughout each of the two syncytia because we said they are separated from each other. And so that sort of begs the question, how do we get the contraction from the top to the bottom? And that is what we're going to be talking about now, and that is the cardiac conduction system. Now, it ends up, you have a special location in the right atrium that is responsible for generating the heartbeat. And it will generate a heartbeat about 80 beats per minute. And that location is called the SA node. And that stands for Cynoatrial Node. And so what it is it's right in the top of the right atrium, right in there where the superior vena cava enters. So it's real high in the right atrium. And what this little spot does is it spontaneously generates a contraction, as I said, about 80 times per minute. And so when that happens, what we're going to see is that the contraction is going to spread across the two atria, and we're going to get lub. Remember, lub-dub? So we're going to get the atria contracting as a result of the SA node contracting. But we had said the lub doesn't do anything for us if we don't have the dub, right? And so what we have is we have another node that we talk about, and that is called the AV node. And where is it located? Right there between the atria and the ventricles. So hence the name Atrioventricular Node. So it's right there between the two. And what does it do? Well, we have to think about what is the purpose of separating the two contractions. So the purpose is to give time for the atria to pump the blood into the two ventricles. Remember, we had said atria receive blood, and then ventricles pump blood. And so we have to have a little bit of time, and it's when we do EKGs, you'll see it's about 0.2 of a second in many people, let's just say. It's sort of in that range. And so we have this delay. And so what we do, and we'd said this communication between cardiocytes is very, very fast, right? So as soon as the SA node starts to contract, that spreads across to the other atrium, the left atrium, and it contracts approximately at the same time as the right atrium. And we'd said what they're doing is they're filling or pushing the blood into the two ventricles. And so we needed a delay to give us just a little bit of time for that blood to get down there. And we have valves that prevent it from coming back up, right? The atrioventricular valves. So the AV node is putting on the brakes and giving it time to happen. So what happens is the AV node puts on the brakes and then it sends the signal on. And so what the signal does is it goes from the AV node down through the bundle branch, which is in the septum between the two ventricles. And when it gets to the apex of the heart, it then heads back up via Purkinje fibers. So we have the SA node to the AV node to the AV bundle to the bundle branch. So the AV bundle to the bundle branch to Purkinje fibers. And so once the signal gets to the bottom of the heart, it then starts spreading everywhere. It starts the ventricles contract from the bottom up. And so that is how we separate the contraction of the two atria from the contraction of the two ventricles. We have this separation. And the slide I've been talking about that's explaining all of this is right over there. So you can see we have the SA node to the atrioventricular, the SA node through the atrial syncytium down to the AV node, then through the AV bundle, through the bundle branch. And so this is going to the septum. This is where the bundle branch is in the septum between the two ventricles. And then it goes down to the apex of the heart and comes back up via Purkinje fibers. They're very fast conduction pathways. And we end up getting the contraction of the two ventricles separated from the contraction of the two atria. All right, so here we can see on a more realistic heart, you can see right up here, I'd said this is the superior vena cava. We'll look right here. That's where the SA node is. And then down here, right here on the border between the two atria and the two ventricles, we have the AV node. And then we'd have the AV bundle and the bundle branch that go down to the apex of the heart. And then when they start coming back up, it's via Purkinje fibers. And we had said, what does that do? That is going to cause the ventricles to contract. I have some videos. Make sure you click on these links. This is a video that is showing, we had said, we have these atrioventricular valves, and we had said they have cusp. And on the cusp, we see that we had said they're attached via chordae tendineae to papillary muscles. And we had said the papillary muscles pull down when the ventricles are contracting, and that's to prevent the blood from flowing backwards. Well, occasionally those chordae tendineae get broken. and you get what we call a prolapse. And so we have a mitral valve prolapse video for you to look at here as while you study. Make sure you look at it. It's interesting. You'll see backflow. That's what it's preventing. All right, next slide is going to be electrocardiogram. I'm going to draw on the board and we'll come back and start another little video. So we'll be coming back with slide number 16.